Hello viewers, I am going to discuss one very important area of bioelectronics entitled biosensor. This is indeed a multidisciplinary area in the sense that it combines the knowledge of bio and electrochemistry, solid state and surface physics, bioengineering, integrated circuit silicon technology, and data processing. And if we see the present scenario as far as its research activity on this area is concerned, this area has at present generated a great number of publications, a flow that shows no sign of diminishing. It has already affected many frontline fields, including biomedical, agriculture, defense, food safety, and environment, with dominant role in the medical field. In this topic, I am going to discuss about the definition of biosensor, its working principle, along with some characteristics which are required for biosensor development, also including some additional factors that need to be considered during the development process. Let us now define what is called biosensor. A biosensor is an analytical device that incorporates a biological recognition element in direct spatial contact with a transduction element. This integration ensures the rapid and convenient conversion of the biological events to detectable signals. That is important point in case of biosensor is this, that it is an analytical device. That is a quantity which is to be measured will be in a analytical form. And it also has one biological recognition element, generally known as bioreceptor, which is in direct spatial contact with a transduction element, technically which is known as a transducer. So in a biosensor, therefore, the phenomenon is recognized by a sensing biological system called bioreceptor, which identifies the analyte under examination and modifies it with the help of biological components into a product that can be detected by a transducer. And as per International Union of Pure and Applied Chemistry recommendation, biosensor is an independently integrated receptor transducer device capable of providing selective, quantitative or semi-quantitative analytical information using a biological recognition element. And referring to its working principle, biosensor can be thought of a system consisting of two blocks connected in series along or integrated with other components such as an amplifier, processor, and display. The first block is a biochemical recognition system, and the second is a physiochemical transducer. The biochemical recognition system consists of bioreceptor, which can be either biological molecular species like enzymes, proteins, antigens, antibodies, etc., or living biological systems like cells, tissues, plants, etc. The main task of the biological recognition system is to convert the information that is received from the biochemical part, which is an analyte, into a physical or chemical or biochemical response signal. What happens actually? A specific molecular interaction occurs, which thereby changes one or more physicochemical parameters. As a result of this interaction, these changes, electrons, ions, gases, heat, light, etc. are produced. These quantities are then quantified, generally which are converted to an electrical signal with the use of transducer, whose output is fed to the signal processor to give the desired result. A schematic representation of biosensor is shown here. This basically consisting of two blocks. One is this part, this is called bioreceptor, which is in direct contact, spatial contact with some sort of transducer. Transducer must be selected in such a way that the conversion due to the bioreceptors use that can be detected by this transducer. So this is the analyte sample which will come in contact with the bioreceptor. Then the interaction takes place, gives a quantity which is detectable by the transducer. Transducer then converts that signal into a electrical signal that will be then processed, amplified, and finally displayed. So what we have understood from this schematic representation, that biosensor is cannot be directly regarded as a device, but basically it is a system consisting of many parts, consisting of many blocks. So bioreceptors, that is the first part, first block. What type of bioreceptors can be used? And what are the properties of these bioreceptors? So bioreceptors is made of highly specialized molecules with the appropriate specificity and sensitivity. The biocomponents which can be used as bioreceptor are generally enzymes. Enzymes, we know, you know that these are basically biocatalysts that generally speed up biochemical reactions. These are basically proteins that lower the energy threshold at which a given reaction takes place. A large number of enzymes are commercially available, for example, glucose oxidase and urease. Antibodies, also known as immunoreceptors. The antibodies, we all know, because we in our body also some antibodies are generally developed due to the antigens. So these antibodies are generally glycoproteins which are produced by the immune system against specific external substances called antigens. These antibodies also can be used as bioreceptor. Then simply receptor. These are generally most of which generally reside in the biomembrane that separates the internal part of a biological cell from the outer environment. And the regulation of biological processes at the molecular level is based on specialized protein structures called receptors, who is able to recognize a number of physiological signals. A very common example which is generally associated with neurology, the acetylcholine receptor is the best known receptor in the field of neurotransmission. Cells and tissues. If a biosensor makes use of complex reaction schemes, then it may be more convenient to use microorganisms or cells where the metabolic pathway is naturally present. Tissue is also another option that has the advantage of cohesion, has a structure that is robust enough to be attached directly to the transducer. So all these bioreceptors, all these biomolecules can be used as bioreceptor to react with the analyte of interest. And as far as biosensors are concerned, these sensors must possess some characteristics such as selectivity. Selectivity of a biosensor system refers to the extent to which a bioreceptor can discriminate a particular analyte of interest under given conditions in mixers, simple or complex whatever be, without interference from other components in the mixture. Specificity, on the other hand, refers to the ultimate selectivity, that is 100% selectivity and 0% interference. This means that the bioreceptor is specific to a particular analyte of interest, recognizing no other. An example is given here showing the selectivity graph of an acetylcholine sensor. Here, experiment to know the selectivity of the interference effects of other biomolecules. Some sort of experiment must be conducted in laboratory. Here, suppose in case of this example, in case of suppose acetylcholine sensor, first one experiment is conducted which has only acetylcholine, and in this experiment the output is measured, which is shown as IDS. So during this simple pure acetylcholine analyte measurement, this amount of reading has been obtained, for example. Now with that analyte, one urea with some sort of concentration has been mixed, and again it is experiment is conducted to see the output. Here mixing all like this, glucose, uric acid, etc., this experiment is generally conducted, and from this it is seen that actually a sensor cannot be influenced by other molecule. Only it is sensitive to acetylcholine under examination. Another very important term, sensitivity. Sensitivity is the measure of the smallest detectable change in the input. Mathematically, it may be defined as change in the quantity of the sensor caused by the input quantity due to the change in the input quantity being measured. So if we take one simple example, we have earlier discussed EIS structure, electrolyte insulator semiconductor structure. In that case, the detectable quantity by the system is psi naught caused by pH of the solution. That is, psi naught is produced the surface potential due to the changing pH of the analyte under examination. Therefore, sensitivity in case of suppose EIS structure is given by d psi naught by del pH. That is, change in the quantity of the sensor caused by the input quantity, which is surface potential psi naught. And the responsible input quantity which has brought this change is the pH of the solution. So basically sensitivity then del psi naught by del pH. So if we draw one now curve between the surface potential and pH, then naturally, since it is a differential quantity d psi naught by del pH, so the slope of that curve will give the sensitivity. If we want to determine the sensitivity of the sensor, then one experiment must be conducted in the laboratory to find the slope of the curve. That slope will give the sensitivity of the sensor. Another important term, characteristic of the biosensor is limit of detection, which is defined as the lowest quantity of substance that can be detected. Mathematically, limit of detection can be defined as LOD equal to T into sigma divided by S. And also one important thing we should remember, that as far as technical side is concerned, every characteristic terms, say for example selectivity, sensitivity, LOD, etc. This should follow some sort of mathematical expression, or this must be determined. This must have some sort of means or technique to determine these characteristics experimentally. That provision technically must be available. Here in this mathematical expression, sigma is the standard deviation and S is the slope of the curve drawn between the substrate concentration and their responses. And if substrate concentration is supposed to be input, and then input versus output curve is nothing but a output characteristic curve. Tezpur characteristic curve, and therefore naturally from Tezpur characteristic curve of the sensor, we will get the value of S. And indirectly, from the previous discussion, this slope is nothing, but mainly it is basically the sensitivity term, because Tezpur characteristic, that is input output, from the input output relationships, the slope generally gives the sensitivity parameter. And here sigma can be calculated using the equation this, delta equal to this much. Here this x and x bar are the sample value and average value of the samples respectively. And this n is the number of samples to be detected. Repeatability. Actually, as far as fabrication is concerned, we have to perform many tests, because after fabrication of one device, it cannot give guarantee that it can again be fabricated for future use. For that, many tests must be conducted. One of them is repeatability. Repeatability, that is, it deals with how consistent a particular sensor is against itself. It can be used to describe the ability of a sensor to provide the same result under the same environment over and over again. For instance, this figure shows the comparison of 5 tests performed on the same sensor under similar conditions. Here, of course, for the same sensor, I have shown 5 tests and similar results have been obtained. Under these conditions, we can say that the sensor is repeatable. But this test should not be confined only to one sensor that we have fabricated or we have developed. But this test supposed to be conducted at least for 5-6 fabricated devices under similar conditions. Then we will be confirmed. We will be certain that the fabricated sensor or developed sensor is essentially a repeatable one. Then reproducibility. Reproducibility is a characteristic of a sensor which tests whether a fabricated sensor can be mass produced or not, because only one sensor cannot be effectively used for other purposes. So many sensors will be required. So there must be, we must conduct one experiment to see, at least for 5 or 6 devices, that whether it can be mass produced or not. For that, this must be conducted, and basically, generally at least 5 sensors are developed using the same procedure under similar conditions, and their performance in terms of their output characteristics is measured. All sensors are supposed to give similar results as shown here. If we get this type of curve, that is, all devices are capable of giving the same result, then we can go for mass production. Then stability, one of the very important characteristic of sensor that it generally deals with the degree to which sensor characteristics remain constant over time. Changes in stability occur on account of components aging due to changes in ambient conditions, leading to the decrease in sensitivity. Stability as a performance factor defines the shelf life of the biosensor system in question. Here, per month, one example is shown here. Per month, some tests are conducted for the same sensor, for one sensor, and corresponding output are recorded, and per month curves are drawn. Here, of course, up to 6 months, the sensor has shown similar results. Under such circumstances, we can say that sensor is stable for 6 months. That is its lifetime or shelf life is 180 days. Linearity and dynamic range. Linearity, it actually reflects the accuracy of the measured response for a set of measurements with different analyte concentrations to a straight line mathematically represented as y equal to a plus sx, where a is the concentration of the target analyte, y is the output signal, and s is the sensitivity of the biosensor. In this context, the term linear range is used to describe the range of analyte concentration for which the biosensor produces a linear response. For this also, we have to conduct experiment. So here one example is shown where output is measured by varying the reference voltage, and from here to this point it is observed that found to be linear. So from this point to this point at least. However, another time dynamic range of the sensor is also used to describe the maximum and minimum values of the analyte concentration that can be measured. Dynamic range can be linear or non-linear. Another is response time. This refers to the total time taken by the biosensor to produce the output signal, qualitative or quantitative, that can be recorded and subjected to interpretation. And recovery time, it refers to the interval time required by the biosensor between two subsequent sample analyses. It is desirable to have as low a recovery time as possible. Usually, a recovery time of within few minutes is desirable. Also, some additional factors or features for successful commercial applications are required, such as for in vivo applications, biosensor probe should be tiny, having no toxic or antigenic effects. Biosensor or its sensing part should be sterilizable without destroying its function for monitoring an analyte in the fermentation setup. Complete biosensor should be inexpensive, small, portable, and capable of being used by semi-skilled operators. And finally, there should be a market for the developed biosensor. It should also fulfill the legislative and other statutory requirements for the country. And some also related to it cofactors and enzymes involved in the biorecognition recognition reaction. If these are not avidly associated with the biocatalyst, then this should be preferably co-immobilized with the biocatalyst to reduce the operational steps involved in the practical use of the biosensor. With this, I conclude this topic. Thank you.